We now have Maartje Huijbers from the Department of Neurology at LUMC in Leiden, and her title of her talk is The Peculiar Forms of IgG4 MuSK Antibodies and How They Can Be Used as Therapeutics.
Thank you. All right, so good morning, everyone. Of course, I would also like to thank the organizers for inviting me to come and work here. Didn't always feel like that. I'm very much enjoying the stay here, and not only for the fun, but also for the science. And also, I would like to congratulate the organizing committee, because this is a tremendous amount of work, and I think you did really well. It's also nice this is broadcasted online, because now I know my husband can actually watch me online and see that I'm actually working. So hi, Paul. This is me working. Today I'd like to take you on the journey we had. We usually do research towards autoimmune diseases of the neuromuscular synapse, and in particular the pathophysiology, and this is one of the stories or research lines we've been following, and I will take you through it, but here we basically looked into the pathophysiology of a disease and ended up starting to develop a first-in-class antibody treatment for neuromuscular diseases. So these are my disclosures, since I'm in academia. First a bit of background on the disease that I work on. So I work on myasthenia gravis, and literally translated from Latin, this means severe muscle weakness. And this is also how these patients present. So they have, in fact, fluctuating muscle weakness. Can be rather mild from this specific hanging of the eyelids. It's called ptosis, and it's usually on one side, very typical for myasthenia gravis. It can then progress and basically affect every single skeletal muscle in the human body, including those for speaking, breathing, walking, etc. It's been known since the 70s already that this disease is caused by autoantibodies directed against key proteins at the neuromuscular synapse. So here you can see in pink or purple these muscle fibers running alongside of each other, and a motor neuron entering and then branching off to form contact points with every single muscle fiber. So every muscle fiber has one single contact point, and that is what we call the neuromuscular synapse. So in a subgroup of myasthenia patients, the dominant autoantibodies are directed against muscle-specific kinase, or in short, MuSK. And what was rather surprising was that the clinicians observed that disease severity in these patients correlated with the amount of IgG4 MuSK antibodies. And that, of course, raised a lot of discussion. There was even an editorial written in Neurology, a very prominent journal in our field, where they said, well, it's very nice that they found these antibodies, but they cannot be pathogenic because they're IgG4, and we all know IgG4 is virtually incapable of activating complement, so they're not pathogenic. So this is actually where I started my PhD, to answer the question, can IgG4 MuSK antibodies actually induce myasthenia gravis? So to answer that question, we collected plasmapheresis material from these patients, which is often given as a treatment when these patients experience a crisis, so they need to get ventilatory support. And then we quickly wash out these antibodies, collect the plasma, purify the antibodies from it. And we can do that selectively these days, so we can purify the IgG4 fraction or the Ig123 fraction from the exact same patient. And we injected that in not-skid mice. And what will happen, and I'll show you in a minute in this video. What's important is that the mice carry markings on their tail, so the more markings, the higher the dose. And red means IgG4, green means the Ig123 fraction, the highest Ig123 fraction from the same donor. And this is what happened. So you can see that this mouse is starting to develop, is virtually incapable of moving around. It received the highest dose of IgG4 and is clearly showing myasthenic muscle weakness. This mouse received the second highest dose. It's a little bit more capable of moving around. And this one is even doing a bit better, but still showing this typical kyphosis and also muscle weakness upon testing. And if you look at this happy fellow that runs around, this is the control. Well, that mice received the highest dose of IG123 from the same patient. So from these experiments, we concluded that IgG4 on its own, without the presence of any other immune system component, no B cells, T cells, no complement, is sufficient to induce the disease, and it does so in a dose-dependent manner. And well, this video summarizes it. We had lots of different outcome measures to prove it, but I think this says it all. So of course we wanted to understand how do these antibodies do this. And if you open up the animal and look at the muscles, you can see the neuromuscular synapses. They usually look like these nice pretzels. It's like a fingerprint. Every muscle, every synapse is different, is unique, and they form these pretzel-like shapes. But in an animal exposed to musk IgG, well, this structure is completely obliterated, basically. And also, if you look at the cross sections here, then here you see a nice synapse curved around the muscle fiber, and here you can see the receptors that mark the synapse move away from the structure, from the cell surface, and move internally. So, of course, next we wanted to figure out how do these antibodies cause the destruction of the synapse. And to explain it to you guys, I need to give you a little bit of background on how a neuromuscular synapse is organized. So you probably all know the motor nerve terminal and the muscle fiber. And the way that neurotransmission takes place is through acetylcholine. It's the neurotransmitter. It's being released by the motor nerve terminal. And the signal is received by acetylcholine receptors on the muscle fiber membrane, which then, if sufficient receptors are activated, can cause muscle fiber contraction. However, most people do not know the second important signaling cascade, which is more of a trophic one, where the motor nerve terminal releases a substance called agrin, and agrin then binds a protein called LRP4, which is tethered to the membrane here, which then can interact with MuSK, and MuSK is a receptor tyrosine kinase, so it needs to dimerize to become an active kinase, and can then signal intracellularly, activate all sorts of pathways, but one of them culminates in the clustering of these receptors. And we know that proper clustering of these receptors, while opposing the release sites of acetylcholine, is very critical for efficient neuromuscular transmission. So in those years, we also mapped the dominant epitope of these MuSK autoantibodies in the patients, and we found that it was actually that all patients had a dominant epitope in the N-terminal IG1 domain. And around the same time, a colleague of ours, Steve Burden at NYU, he showed that the N-terminal IG1 domain of MuSK is critical for the interaction with LRP4. So logically, we reasoned that these patient antibodies may cause myasthenia by obstructing this interaction. So I actually went to New York, and this is one of these occasions where you do an experiment, and within 1 month you actually know the full story. It actually worked. I know this is probably once in a lifetime, but within a month, we had the evidence that that's exactly what happens. So these antibodies, they obstruct the interaction between MuSK and LRP4. They do not cause internalization, and they do not block the dimerization of the kinase. But by obstructing this activating signal, they prevent the activation of the kinase, and therefore there's a loss of acetylcholine receptor clustering signal, and that ultimately leads to muscle weakness. And this paradigm has now been established by several groups, and it's really, I guess, established also in the field. However, I drew this picture wrong in the first time, because there's something special about IG4. IG4 is not like any other antibody. It actually looks like, it actually has several peculiar features. And so the first thing that IG4 can do is that it interacts with other IgGs through its heavy chain. Now, I don't think we actually know what the role for this is and how relevant it is. We do know that it sometimes can impact your assays, plate-based assays, for example. The second thing is that it can undergo FOB arm exchange, and I'll explain that a little bit in a minute. But what it effectively comes down to is that an IG4 is effectively produced as a bivalent molecule, but once it enters circulation, it becomes functionally bivalent, of a bispecific and functionally monovalent. And because of this, it does not form these antibody-antigen complexes. It binds in a monovalent fashion. So what does that look like when you think about these MuSK autoantibodies? Well, then the plasma cell produces a MuSK antibody like any other antibody in a bivalent fashion, but as soon as it reaches circulation, due to the large heterogeneity of the IG4 pool in circulation and FAP arm exchange, the vast majority turns out to be bispecific and monovalent in the circulation. And we actually also proved, together with a group in Maastricht, that this is really the case, that 99% of these MuSK antibodies are actually functionally monovalent. And the reason for that is two residues that are different in the constant region of the IgG4 that prefer intrachange sulfide bridges over interchange sulfide bridges. So these IG4 molecules, they're continuously moving around, attaching to other IG4s, and that's how this comes about. So I should have drawn this picture like this. And now the next research question we wanted to answer is, does it really matter that these antibodies are of the IG4 subclass and that they are becoming functionally monovalent? In other words, does it matter whether you have 2 or 1 binding arm for MuSK in the pathophysiology of MuSK-MG? So to answer that question, of course, we need to have a pure source of these antibodies. So as others also shown, there's a nice way of isolating autoreactive B cells from PBMCs from patients. So we collected those, and this is work, by the way, from my long-term colleague Yvonne, who did all of these experiments, and this slide sums up 2.5 years of work. So what she did was she set up a method to selectively purify these MuSK-specific B cells. We sorted them in single-cell cultures. We differentiated them to plasma cells, and then we measured in the medium the anti-MuSK reactivity. And from the positive clones, we isolated the transcriptome and the BCR sequences. And sure enough, from, for example, this patient, we were able to isolate 5 different clones. Surprisingly, they were not all IG4, but they were of different subclasses. They also made use of different VDJs, although there's some overlap that can be seen. They were highly mutated, suggesting that they had undergone affinity maturation. 2 clones contained N-link glycosylation motif in their variable domain. And they all bound the main immunogenic region on MuSK, so the IG1 domain. So with these in hand, we could address this question. But of course, we also had to make these stable bispecific antibodies, basically. So what we did there was we adopted the controlled FAP arm exchange generated by Are Labrein in GemMARP, and we adjusted it to work for IG4, basically. So what you do then is that you have the IG4 MuSK antibody with the S228B and an irrelevant donor antibody, in this case a B12 antibody, which has matching mutations. And basically, if you then combine them in a reducing environment, they favor the interaction with one another. And this is a directional reaction, I guess, that ends up with these bispecific antibodies, and after they have connected with one another, they favor this hetero-interaction, so they do not let go anymore. And if you then do that in the excess of B12 antibodies, you can drive this process basically in such a way that you get left with over 99% pure bispecifics. So here you see the evidence from that, the mass spectra of the MuSK clone, the intermediate and the irrelevant donor. And we also set up a method to actually quantify the purity of these monovalent antibodies, because we reasoned that if these fractions would not be pure, that they may cause a bias. So here you can see the quantification, and you see that there's virtually no bivalent parental clone left in these bispecific antibody pools. So with these antibodies in hand, we went back to the not-skid mice and injected them every 3 days with different dosage. And just to highlight, if you're lost during my presentation, I've highlighted which type of antibody I've used in the next couple of slides. So here we wanted to compare these bivalent ones versus the monovalent ones. And to cut a long story short, this is what happened. It's really a black and white picture. Mice exposed to these functionally monovalent MuSK antibodies, they develop progressive myasthenic muscle weakness, for example, shown here with loss of grip strength and hanging time, but also with the body weight. And the bivalent antibodies, well, these mice actually did not show a phenotype, perhaps except for this 3b5 clone, where we saw maybe a little bit of myasthenic symptoms. So we concluded from that indeed the monovalent MuSK antibodies are much more pathogenic than the bivalent ones. Next, of course, we wanted to see if these bivalent MuSK antibodies then don't have any effect at all. So we repeated the experiment, but just expanded the exposure time and also increased the dosage. And then this is what we found. So this is this 3B5 clone where we already saw a hint. So in the end, it did progress and cause and develop myasthenia, but at a much slower pace. So we think there's a completely different mechanism underlying this pathophysiology. Whereas for the 3F6 clone, we never were able to find a phenotype, and we continued dosing in other experiments for much longer, but this clone did not induce myasthenia. So there's a clone-dependent pathogenicity for the bivalent ones. So how can we explain this clear difference between the monovalent and the bivalent antibodies? So to answer that question, we went to the C2, C12 mouse myotubes. These are cells that contain all necessary machinery to mimic a muscle fiber membrane, basically, except for agrin, because agrin is released by the motor neuron. So we just add that to the dish, basically. And then you can follow the differentiation and sort of the clustering of acetylcholine receptors. And when you do that, here you see we've precipitated for MuSK, and you can activate the kinase if you add agrin. So here we monitor phosphorylation of the kinase. The same is true if you add in a control antibody, then there's regular activation of agrin and not in the absence of agrin. And then we saw something peculiar happening when we added these antibodies in the absence of agrin. There was nothing going on with the monovalent ones, but the bivalent ones actually activated the MuSK kinase itself. In the presence of agrin, we saw that the monovalent one didn't show any activation of the kinase, whereas the bivalent one still gave activation of the kinase. And we saw the exact same for other clones. So from this we concluded that in fact these monovalent antibodies, they act as antagonists. They block this interaction between MuSK and LRP4 and cause the disease. Whereas the bivalent MuSK antibodies, they can actually act as agonists and force the dimerization of the kinase. So from that we came to this sort of summarizing outline with this Ig4 MuSK antibodies being produced. They rapidly become functionally monovalent in patient circulation. The monovalent ones, they obstruct this MuSK-LRP4 interaction. There's a loss of signaling, a loss of clustering. Therefore, they are highly pathogenic, and these mice tip over within, 5 to 10 days. Whereas if you have a bivalent MuSK antibody against this Ig1 domain, it's actually clone-dependent pathogenicity. Some of them, they're actually able to induce acetylcholine receptor clustering. They're not pathogenic. They do not induce a phenotype. Whereas others do result in a, as yet unknown fashion, in acetylcholine receptor declustering. And they cause myasthenia, but with a much slower onset and progression. And then we had an idea, because we can use this knowledge to treat neuromuscular diseases. Because there are quite a few neuromuscular diseases that are hallmarked by impaired neuromuscular transmission. And as I showed you, this MuSK is really a key hub for stabilizing the structure. If we can use these MuSK agonists to stabilize the structures, the synapses, in diseases where neuromuscular transmission is impaired, that may be beneficial for them. So this is where we wrote a patent saying that we could apply them to treat neuromuscular diseases like ALS, myasthenia gravis, SMA, etc. And then on the other hand, this is also a cool idea, because there are also neuromuscular diseases that are hallmarked by hyperactivity of muscle. And often cases in these patients, Botox is, for example, used to impair neuromuscular transmission or limit it so that the muscle tension is released a little bit. So this could be a Botox alternative, basically. So this is the other, we call it the inhibiting patent. So the rest of my talk, I will focus on this because that's where most of our efforts gone these days. So we reached out to argenx, and they were very interested in pursuing this idea. So the rest of this work is done in collaboration with them. And especially I would like to mention Jamie, who's very great postdoc who did all this work. So what we did was we continued with our monoclonal antibodies from the patients, or some new antibodies raised against the same domain. We optimized and de-risked them, and then yeah, we tested their ability to activate the kinase, for example. So here you can see a plot. We put the natural ligand agrin at 100%, and then we tested the different clones. And when they have a similar color, they basically derive from the same clone. So these are different variants, basically. And as you can see, most of these clones, they are able to reach about 100% of musk phosphorylation, like the natural ligand. Some of them, even surprisingly, they can turn musk on more, if you were, yeah, if you can say something like that. So, but yeah, they were definitely a good agonist. We also looked at acetylcholine receptor clustering, and this was quite interesting because apparently turning on the musk kinase is not the same as inducing acetylcholine receptor clustering, because here, if you look at the 100% line of agrin, they are a bit less capable of reaching this threshold. For some, it's up to 90%. But they're a bit less successful. So there's really a trick that agrin is doing that is very hard to mimic with these monoclonal antibodies. And we also looked at the sizes of these clusters that form then on the myotubes. And what was striking was that these agonist antibodies, they induce more smaller clusters, whereas agrin is able to induce more larger clusters. So to us, very interesting, but I'm not sure it's very relevant for the rest of this story. But what is very nice is that we can use this model also to preclinically test basically the therapeutic idea of using this for musk myasthenia gravis. So to do that, we add these monoclonal antibodies, the monovalent antibodies, or polyclonal patient IgG4 to these myotubes, and then half an hour later we treat them with the agonist and then monitor the formation of these clusters. And what we can show here to summarize is that the different clones have different abilities of rescuing basically this in vitro myasthenia model up to like 40 to 50%. And you have to realize it's a very radical model, right? Because there's a huge amount of patient antibodies already in the dish inhibiting these myotubes, and then still these agonists are able to overcome this. So this to us was very promising, and we moved ahead with the animal experiments. And I like to show this slide because I do think it's good to also tell, you know, the young scientists around among us that not everything flies in one go. And this is definitely an example of that, because we just wanted to make sure that these musk agonists were not inducing any myasthenia in any way. So they were injected in mice, and all of a sudden, in black 6 mice, all of a sudden, male black 6 mice started dying. There was a sudden death. It was not related to any myasthenia. Here you can see the body weight was really stable, but one after another would just drop dead. And when we opened up these animals, sorry for the bloody picture, but yeah, we could see these expanded bladders. They were bloody. And what we found was that these mice were suffering from mouse urologic syndrome. It's a situation that's been described before in other preclinical tests, I guess, where the animals probably have difficulties with ejaculation. Therefore, the semen is stuck in the urogenital tract, basically, and then the urine cannot be released anymore. And yeah, well, you can imagine that's not a happy ending. So yeah, I think we invested like close to a year to understand how this pathology came about. There's no way or association by any sort from the clinic or that we could explain this, and we still have no idea. So if you do, please come and tell me. But yeah, that's the story. However, fortunately, while we were developing these antibodies, we started teaming up again with Steve Burden from NYU, because he had a similar approach in mind, just targeting a different part of musk. So we continued this 3-way collaboration, and once we saw these problems with these Ig-like 1 domain antibodies, we shifted gear and started working on Frizzle domain antibodies, which are also directed against musk, but a different part. And well, to cut a long story short, I'm also not allowed to show you everything. It's all unpublished, but this is too excited not to show you. So what we did is develop in a similar fashion in the end a musk frizzle domain antibody. And again, we went back to our animal model. So this is the mouse model I showed you in the beginning, where we inject polyclonal patient IgG4 to induce myasthenia, but then we add in this agonist musk antibody, and this is what happened. So if you do that with a control antibody, these mice start to die rather rapidly, but in the presence of this MuSK agonist binding the frizzle domain, yeah, virtually all mice survived. And you have to realize that every day we inject these mice with patient antibodies, so this is really a radical model. If we look at grip strength and inverted mesh hanging time, the rescue is also very surprising for this particular patient. So yeah, we're very excited about these results, and this is one of the reasons why I started phase 1 clinical trials this spring with this antibody. So of course you may wonder, well, how relevant is it? MuSK myasthenia, super rare disease. But in fact, yeah, we hope that we can apply it to other neuromuscular diseases as well, as I explained. So here you see an example of a study by Steve's group, where they used the SOT1 ALS mouse model to model ALS basically. And then they added in a MuSK antibody directed at the frizzle domain. This is MAP13 in this case. And you see, for example, if you look at I think this is denervation or innervation, and this is denervation, that there's much more innervated synapses and that function is improved in these animals. However, it does not last until the end, because the endogenous defect that's in these motor neurons, yeah, that's not rescued with this, but the idea is that you would be able to maintain muscle function much longer, at least in these patients. And this is what we saw, what they saw in these mice. This is another, I think, spectacular example. This is a DOC7 congenital myasthenia syndrome. Usually these animals die within 2 weeks after birth. But what they show with a different MuSK antibody called X17 is that if these animals are exposed early in life, that they actually live on to age. I think they went way over 100 days now. And you can also see that the synapses are rescued significantly. So these are exciting things for us, I guess. So my conclusions for you, I hope I've convinced you that patient IgG, IgG4 in general is not an innocent bystander. It's really context dependent. And in the case of MuSK myasthenia gravis, it can be very pathogenic and induce a dose-dependent muscle weakness. This is caused by the fact that these antibodies obstruct MuSK-LRP4 interaction, and therefore there's a loss of acetylcholine receptor clustering. So the functional valency of these MuSK antibodies is key, because the monovalent ones are antagonists, the bivalent ones are agonists. And thus for the disease mechanism in these patients, isotype switching, this class switching to IG4 and the FAP arm exchange and affinity maturation, they're all key steps in developing this disease. I've shown you that IgG1 domain MuSK antibodies cause sudden death in male black 6 mice. We did not observe this, by the way, in not-skid mice or in any of the females, so still very puzzling. And that frizzle domain MuSK antibodies seem to be very promising therapeutics for neuromuscular diseases like MuSK-MG. And I hope I have also convinced you that doing this in-depth studies towards the pathophysiology of these diseases can be very helpful in developing getting ideas for novel therapeutics. And with that, I would like to thank my group at the LUMC, in particular Jamie, Dana, Roy, and Yvonne, who did most of the work I show you today. Also my long-term collaborator Steve and Jan, Martijn, the 2 neurologists who see all these patients, and Jaap and Silver for long-term collaborations. All the patients, the funders, of course, and you for your attention.